Okay, so hi everyone. Welcome to Bio 285. This is our first week review. I am Emily. I am the TA running this session. A lot of times there are going to be two TAs, but today was just a busy day for people. This is my second semester, so I've run these reviews before. And we're just going to get started. Feel free to stop me if you have any questions, and we will also have time for questions at the end. These slides are not available on Moodle. Good question, but we do send out the slides after the review session. So there will be an email with them. Okay. So this week we talked about amino acids and level of protein structure. So that's what we're going to get into today. So first we're going to start with amino acid structure. So these are beautiful pictures for different ways to visualize, because I know everyone's a different visual learner, but the general amino acid structure is our amino group, carboxyl group, and R group. So I'm going to use this one down here. Can you see my cursor? Can we unmute or in the chat say yes or no?
Yes.
Okay, thank you. So right here, we have our central carbon. Our central carbon is what all of our groups are attached to. So we have our central carbon, also called our alpha carbon, and we have a hydrogen attached to that. That never changes. And then on either side, we have our amino group. Our amino group is our nitrogen, and our carboxyl group, which is our carbon and oxygens, and those are named after the groups they are, an amine group and a carboxyl group, if you remember from chemistry. And those are mostly the same. If they're on the ends, they're charged, and if they're in the middle, they're involved in peptide bonds, which we'll get into. The thing that really makes amino acids different, because we have multiple amino acids, is there are R groups, and we will get more into that, but R groups is how they differ. Everything else is the same. And yeah, this, if you take biochemistry, you're going to learn all about ionization, different pHs, and when things are charged and when things aren't. We're not going to get into that in this class, but for those of you who need to take Biochem 320, you will definitely get familiar with that. Do we have any questions on our amino acid structure? Okay, so we're going to move on to formation of the peptide bond. So if we remember from class and lecture, the peptide bond is the bond between the carboxyl carbon of one amino acid and the nitrogen of the other group. I'm so sorry, I just lost my train of thought. Yeah, so the carboxyl group and the nitrogen, between them is where the peptide bond is. So this is a great question, because a lot of times Professor Francis will ask you where the peptide bond is, and sometimes people get confused and put it between this nitrogen and this carbon, but the carbon that is attached to the R group is never involved in the peptide bond, because that is the alpha carbon, and that's already attached to 4 groups. So the peptide bond only occurs through a dehydration reaction, which means we make water. So this OH and the H end up making water as a byproduct, and we create a bond there. And that's how we attach our amino acids and how we make proteins, because that's how we make our polypeptide, which then turns into a protein most of the time in this class. Yeah.
I have a quick question. Can any amino acids bond together to make the peptide bond? Because that was one of the questions on the homework. It said draw a picture of 2 amino acids separate and then together. So
yeah,
any 2 amino acids, I think I was making it harder than it had to be in my head. So any amino acid can combine together, and then the byproduct would be the H2O, or the product would be the H2O, and it would be the same. The bond would pretty much bond in the same place, no matter what.
Yeah, exactly. Because if you remember, the R group is the only thing that is different, and the R groups aren't involved in this. So this is primary structure. Once you get to tertiary structure, which we will get into today in folding, that's where the different amino acids matter. But when it comes to peptide bonds, we're looking at our 2 groups that are the same, and the bond between the carbon and the nitrogen. And that can happen between any amino acid because they both have those groups. So it'll always be the same. And that question, you just have to pick two. The point is just to pick two. So
Okay. I think I was overthinking it. So thank you.
Overthinking is a common theme, so don't worry about it. That is totally reasonable. So yeah, that's a dehydration reaction. And then if we go reverse and we break this bond, then that's hydrolysis reaction, which means you need water, hydrolysis, hydro, water. Obviously, because we have to add back this OH and this H to break it apart. And if we break that bond, it releases a lot of energy. And it also, because it's a covalent bond, to break it requires a lot of energy. It's very strong and it's not just going to be broken very easily. So that's why our primary structure usually is a lot sturdier, because if you heat it up, our covalent bonds don't break. And we'll compare that to our non-covalent bonds in a little bit. Do we have any other questions about peptide bonds? Okay. And then polypeptide anatomy is peptide bonds, amino acids in a line makes a polypeptide. Poly meaning many, so many peptide bonds, we got a polypeptide. And we have our N terminus and our C terminus. Our N terminus is when our nitrogen is exposed, no peptide bond. And our C terminus is when our carboxyl group is exposed. And so we read N to C, just like you read left to right. So when you're numbering your amino acids, you start at the N terminus. So this would be 1, 2, 3, 4. And so that, I know there was a question on the problem set that said if it's the first amino acids, which side will they be on? Well, if it's the first ones, then we read from the N terminus. So we'd be on that side, right? We would be at the M terminus because we always, always, always start at the N terminus when we are numbering or considering the directionality of a polypeptide. And our groups are arranged by chemistry and size. Sterics means literally that two big things, like they can't, if you think about it, it's three-dimensional space. And if you have two three-dimensional things, they can't be in the same space. So we might not always get to see that on paper, but that is in reality, that's steric strain. So the big things don't want to be near each other. They want their own space. So that's what it means by steric strain. And then chemistry, obviously, we know ionic bonds, two negatives would probably be far apart from each other. And so that's how our R groups are arranged, is they each kind of want to be most convenient for themselves. And then our peptide bond has resonance. So we talked about this in class today. This is kind of tricky. I know a lot of people struggle with this in chemistry. You don't need to know much about resonance other than your electrons are kind of shared between, they're not as, they're shared is probably the best way to explain it. So it's more stable. These peptide bonds are single bonds, that's what they're shown as, but because the electrons are shared and they're a little sturdier, they act more like double bonds. And because they act like double bonds, it means that they're stronger and it's harder, things don't rotate around the bond as much. So it's very sturdy. So the molecules aren't, or the R groups and everything, they're not going to be moving around in space a lot because it's a sturdier bond. Because single bonds you can rotate around, double bonds not so much. And that double bond comes from the resonance in the carboxyl group. Like I said, you don't need to really get into the resonance and the chemistry of it. Just know that the resonance is what gives it the double bond characteristic and the strength to not rotate. Are there any questions on that? I know that one's a little tricky. Since the C end in a peptide bond is as sturdy as a double bond, I'm assuming is what the question is. Why is it shorter than a double bond but longer than a single bond? That's an awesome question. So this actually really does have to do with the chemistry of it, is that when you think of bonds are just electrons. So we draw them as a line, right? We draw it as a line connecting something. In reality, it's the distance that the electrons are traveling. And so a double bond has more electrons, they're more tightly compact. This isn't a great way, I'm not a chemist, so I'm not probably explaining this best, but it's because of the space between the two things that are being bonded, that the electrons are connecting, is smaller. And a single bond, they're further away because it's weaker. And you know, stronger attachments are probably closer together. And so this resonance makes it kind of half. It kind of is in the middle. And that's why, although it is a single bond, it's that number of electrons, the resonance with these other extra electrons kind of moving makes it a little shorter, which is why it has those double bond characteristics. And so that's why it's mid-size, and that's what originally how we kind of figured this out about peptide bonds, because they were looking at it and like, wow, this behaves like a double bond, but it's not the length of a single bond and it's not the length of a double bond. So what is it? And that's kind of how we figured it out. So the reason it is that length is because of the resonance. Resonance bonds are kind of different. We even draw them differently in chemistry. We draw a line with dashed lines. And that's, you don't need to know much about the length, but that was just the unique finding that led us to determine that it was resonant. So if there's a question about that, I think there's a question about that. That's why, that's all it's asking is, hey, how can we explain why this one is like in the middle? And the answer is resonance. That's why. Like I said, I went into a little more chemistry there than you need to, and I didn't explain it very well. Your main thing that you need to know is resonance for this class. If you really love that, take more chemistry classes, you'll definitely do plenty of resonance. Does that answer your question? I know I kind of went off on a tangent there, but I just want to make sure that that's clear. Yeah, okay. So I'm going to move on. Feel free to stop me if there's any other questions on this. Oh, protein structure levels. So this is where we get into everyone's favorite questions. So we have 4 levels, and we're going to start with primary. So primary, first level, is just your sequence of amino acids. So this is literally just the connections, the order we're in. So remember, we count from N to C. And so you just start at N, and it's alanine, valine, alanine, lysosine, and it's just that sequence. That is your primary structure, and it's peptide bonds. Super strong. And so that is very important at those covalent bonds, because a lot of times a great question is, okay, if I heat this up, what's going to happen? Is it going to bother primary structure? And the answer is no, because heat, it destabilizes, but it doesn't denature, I believe is the words. So it might break non-covalent bonds, but those covalent bonds are so strong that it takes really aggressive chemicals to break those up in a lab. So the primary structure is very hard to break, and you're usually not going to, and that is just the sequence. So it's not as much of a picture in your head as secondary or tertiary, the folding and everything. Primary is literally, it's like you write out the alphabet, you just put letters in a random order. Well, it's not random in biology, nothing's really random in the cell. It's very controlled. But that is essentially all primary structure is you write down your amino acids in their order, and that's your primary structure. Very hard to break because they're covalent bonds, the peptide bonds, and that is pretty much the meat of that. And then we get on to secondary structure, and this is where it starts to get a little more fun. So secondary structure, your main bonds are hydrogen bonds. So a big thing to know is what bonds can be involved in what level. So primary structure, we have peptide bonds, we have covalent bonds. Peptide bonds are just a type of covalent bond. In secondary, it's hydrogen bonds, and it's hydrogen bonds between the R groups and the backbone. No, sorry, not the R groups, between the amino acid and carboxylic acid groups. So it's your backbone atoms, remember, because those are the ones that don't change. That's where the peptide bonds are. So we have hydrogen bonds between those. And you can think about, why are some alpha helices and 1 beta sheets? Like, why don't they all fold the same? They're all the same groups. That has to do with your R groups. Because remember, our R groups, when they're in that primary structure, they kind of face away from each other based on their chemistry and their size. And so the way that it folds in a certain, if they're aligned a certain way, when you make those bonds between your amino group and your carboxylic group or your carboxyl group, you end up with a helix, because the helix, the R groups are facing outwards. You can imagine if you have a coil those big groups can't fit inside. So that's a good way to remember it, is that your R groups can't be inside because they're too big. They don't fit in the coil. That's where your hydrogen bonds are. Versus your beta sheet, this is trying to show three-dimensionally, but your beta sheet like this, you have an R group up and an R group down, an R group up and an R group down. So they alternate. And so they're more on the outside. So those are the two secondary structures. This slide is great because it goes into your dipole movement and why those hydrogen bonds exist. Those are non-covalent bonds, that's important. So they're easier to break than your covalent bonds. But I wouldn't focus too much on exactly like the dipole movement between the amino and carboxyl groups. Just know that hydrogen bonds happen because of dipoles, which is differences in electronegativity, which I think we're going to get a little more into when we're talking about tertiary structure. That's what causes the hydrogen bonds, but the big thing is what the bonds are, that you know alpha helices and beta sheets, and that you know where the R groups are. We'll also get into modeling, and this is where your ribbon model that we love. So do we have any questions on that? Okay. So then your tertiary structure, this is your overall shape of your protein. This is a single polypeptide. So we have one polypeptide, one chain, and the way it folds is its tertiary structure. So we had a line, and then we had some alpha helices and beta sheets, and then it folds into the cute little clumps that you see the pictures of in any bio anything ever. Those cute little clumps and those unique shapes that all kind of look the same are due to their tertiary structure. And these are your interactions between your R groups. So all your R groups are either nonpolar, polar, or charged, basic or acidic. Charged are technically just superpolar, but that's how we break them up. So we can either have hydrophobic interactions or van der Waals. Those happen between nonpolar groups. So nonpolar groups just kind of hang out together. They don't want to interact with anything else. They're non-social, nonpolar, non-social. They hang out with each other and no one else. Ionic bonds happen between ions. So ionic bonds are happening between your charged group, your acidics and your basics, or your positives and your negatives. Hydrogen bonds, which are happening between polar, because of the electronegativity difference. And then disulfide bridges are very, very unique and only happen between cysteines. Yeah, I can explain what van der Waals is. Let me just finish disulfide bridges and then I'll explain what van der Waals is. So this disulfide linkage down here is just disulfide to, what is it? Sulfurs. Wow, I forgot the name of the atom. Two sulfurs, it makes a covalent bond. So technically there is a covalent bond in tertiary structure, but like I said, that's rare and it's between cysteines only. So while it happens, it doesn't happen a lot. And it is the strongest of all the tertiaries because everything else is non-covalent. So hydrophobic interaction and van der Waals, there's a slash there because those are essentially the same thing. In some classes, you'll hear it called hydrostatic interactions. And that's essentially, if you remember, in three-dimensional space, things just bump into each other. Always, things are moving electrons, atoms, everything's just moving and things just bump into each other. And those random instances of things bumping into each other are called van der Waals interactions. They're interactions that things are happening, that things are having just because they're near each other and they're hitting each other. So say in a chemistry-based course, you throw a bunch of stuff into a test tube and you're like, hey, what interactions are happening? Van der Waals are always happening because things are just hitting each other. The same thing here is that hydrophobic R groups, amino acids, they don't want to be touching water, which is most of the cell, and they don't want to deal with other polar or ionic groups. So they kind of go find things that are like them, and they kind of just hang out. So they're kind of bumping into each other and just hanging out and clumping around each other, and that's van der Waals. So it's not really a it's just an interaction more than an actual bond. And that's why we don't call it a bond, because it's very transient and it's just happening. Mostly it's a kind of complex thing to wrap your head around, but it's just really mostly important to know that that's happening between non-polar groups because they don't have electronegativity, so they're not really sharing electrons, they're not really bonding to other things. Does that make sense? Does that help answer the question? Did I clear that up? Okay, awesome. So let's move on. Or could a positively charged R group have an ion dipole interaction? Yes. So ion means charged, dipole means partial charge. So like OH, the O is slightly negative and the H is slightly positive. And so that's what happens with hydrogen bonds. An ion dipole means that there's a part of the ionic amino acid that can bond with the slightly negative. So for instance, I'm trying to explain this without drawing it out, which is kind of hard, but you can have an ionic R group that also has a hydrogen that's slightly positive and can form a hydrogen bond with polar group. So even though you have that ion there, it's not making the ionic bond. There's something else in that group, because obviously if it's ionic, it is polar all around, you know, those electrons, that's really affecting the electrons. So when it says ion dipole, it's not necessarily one of these specific things happening, but it's when you have an ionic R group charged and a polar one that are making more of a hydrogen bond. Just because the ionic is also polar, it's just a different part. It's not the charge that's making the ionic bond, it's a different part of it, like a hydrogen that's making that bond instead. So ions can make bonds with dipoles. Usually in the questions you're going to get asked in this class, it's going to be more label them with these bond names. And so in that case, you'd be like, oh, well, it can make a hydrogen bond. You won't have to say ion dipole necessarily, but to recognize, oh, hey, there's a hydrogen there that might be able to actually bond with that dipole, as opposed to just having things having to just be charged. So it doesn't make an ionic bond, is what I'm saying, but they can interact. Does that make sense? I know that's kind of hard to understand, but did that help? Okay. This is going back a little, but I was struggling with the question about the problem set, whether changing primary structure would also alter secondary structure. This is a great question. Normally I save problem set questions till the end, but this is a very good general question. And the way I like to look at it is a pyramid. So say you're building, say you have Jenga blocks, or I don't know what we used to use. You have Jenga blocks, but it's just one after the other. So you have primary, secondary, tertiary, quaternary. If you pull out that bottom block, it's all going to topple, right? But if you take off the top block, the rest might stay. So if you mess up primary structure, it's going to mess up everything else. But if you mess up quaternary structure, everything else can kind of still stay the same because those things aren't being affected. Primary structure determines secondary structure, which determines tertiary structure. But even if you miss a group between or break a bond between R groups, that might not necessarily change the fact that it's an alpha helix. So that's the best way to look at it, is building something up. If you break something below it, everything's going to be destroyed. Because if you break bonds, then your protein can't fold correctly. If you break those covalent bonds and you don't have the same polypeptide, it's not going to fold the same. But if I unravel all my R groups and I pull my thread, my polypeptide, my primary structure is still the same because I didn't break those covalent bonds. Does that make sense as to the directionality there, kind of? And it's not a problem. I know, I like to give people that are here the credit for being here. And so they get more help on the problem sets. That's the only reason I normally save them till the end. So it's not something personal. I just will probably stop recording as an incentive to come to the review sessions. Okay, so we're move on from tertiary and onto quaternary. So quaternary structure is when you have two subunits, also known as two or more polypeptides, that are interacting with each other. And they make all the same interactions as the tertiary, because quaternary and tertiary are the same thing. It's R groups interacting. Quaternary is just when you are interacting between polypeptides. So hemoglobin is a very important protein for carrying oxygen throughout the body. Did a project on it junior year of high school. So I'm very interested in hemoglobin. It's awesome. And so as you can see here, we have 4 subunits. So it's a 4 subunit, 4 polypeptide protein. And so these subunits essentially are being held together through the same bonds that we had in tertiary structure. It's just happening between 2 subunits. That's the only difference. So while every protein is going to have a tertiary structure, if it's formed properly, not every protein has a quaternary structure, because sometimes one polypeptide is enough. It just depends on the protein and what it needs to do. Does that make sense? Cool. More on tertiary and quaternary structure. Like I just said, tertiary is a single polypeptide and quaternary is multiple, usually. Actually, there's just not a usually. I don't know if I've ever seen more than 4, but they exist. It's just not the examples we use probably necessarily, but there's some pretty complex proteins. And like we said, these are hydrogen bonds, electrostatic interactions, which are the same as van der Waals, ionic, and those disulfide bridges. Those aren't all listed, but they're not the same thing, but they are happening between subunits versus just between the R groups in the same polypeptide. I don't want to keep harping on this because I don't want to confuse anyone, so we can continue. We can go back to this if anyone's struggling, but I'm going to move forward for now because I feel like I've spent a lot of time talking about tertiary and quaternary. Domains versus subunits. This is something I really struggled on when I took this class. Subunit is a polypeptide. So if I have 1 polypeptide, I have 1 subunit. If I have 2 polypeptides, I have 2 subunits. A subunit is just the polypeptide folded the structure. So that's the easier one to remember. A domain is a part of a protein that you can find in many different proteins that is folded similarly. So if you remember, structure leads to function. That's like the basis of protein and a lot of biology, is that structure leads to function. So a domain is a part of a protein, part of a polypeptide that has the same structure or similar structures and serve similar functions. So probably giving an example is going to help here. So you're going to learn a lot about enzymes such as kinases. Kinases phosphorylate, they put phosphates on things. So they usually activate or inactivate. So a lot of times we have a kinase, which is an enzyme that puts a phosphate on something. Kinases are gonna have similar domains if they're serving a similar function. The proteins don't all look the same, but say there's a certain pocket that actually, or that it sticks the phosphate on. So how does it know where to stick a phosphate, right? It will just stick phosphates to everything. Well, it has one region that looks the same every time it attaches a phosphate to it. That region would be called a domain because it can be seen on multiple different things, but it always serves the same purpose. So a subunit can have multiple domains. This is definitely a harder concept to grasp, but just think about it as it's part of a protein that has similar structures across multiple proteins that all serve the same function. So no matter where it is, like where you find this domain, it's doing the same thing, no matter what protein it's in. Does that make sense? The difference between them? They're not, the thing is, we compare them, but they're not, like we do this versus this, but they're not really that similar. Subunit, the same motif. I'm gonna be honest, off the top of my head, I cannot remember. If anyone else remembers that off the top of their head, please let me know. If not, I will look that up. I just don't want to leave my screen right now or everyone's going to see all the stuff that I have on my Google Docs right now, or my Google Chrome right now, and that's kind of embarrassing. So I will definitely look that up. So I'm going to keep that question if no one knows. Is a subunit the same as a motif? I think you're right. It's just been a while since I heard the word motif. We don't use it a lot. So I just don't want to give you 100% until I look it up. So I will definitely come back to that and look it up once we get through the slides, which we're getting pretty far on. Okay, so I will definitely come back to that. And if I don't, please remind me. So this is another cute graphic. Shout out to Michelle, who was a TA for 5 semesters. She graduated, but she made these slides and she's amazing and we miss her dearly, but she is so good at finding pictures. So tertiary level is this one train, it's the one polypeptide, but then you have your choo-choo train that has all the different attachments, and that's quaternary. So you got 2 or more polypeptides or subunits. This would be quaternary and this would be tertiary. And I don't know if that helps you visualize better, but it's cute. So. R group interactions, bonds, and more amino acids, etc. So this is what we're talking about when talking about R group interactions. So this is the meat and bones of this section. So here's the beautiful graphic that I'm glad that we can finally see here. With your nonpolar, polar charged, positive and negatively charged amino acids. So you will always have this chart on all exams. So you do not need to memorize these. If you're going to med school, you do have to memorize these. So maybe get a jump start on that if you're pre-med. But if you're not going to medical school and it's just for this class, do not worry about memorizing these. And this basically just lists out what the different R group interactions can be. So charged groups can make ionic bonds with each other. So a positive can make a bond, an ionic bond with a negative. Obviously, we all know that opposites attract. And likes repel in the case of ions. So 2 positives will not be making an ionic bond. They're going to repel each other because they hate each other. Polar uncharged R groups, which is our blue group down here, we make hydrogen bonds. Hydrophilic means we like water. So your polar and your ionic, they like water. So they're cool to make hydrogen bonds. We love it. And yeah, so when it says hydrophilic, and it's talking about these bonds, the CO, CN, that's what's actually in the R group, the COs, the CNs. So anytime you see an O and H's kind of together, that's going to be polar because they have electronegativity versus C's and H's, which have the same electronegativity. So there's no difference. And so that's really why it makes it hydrophilic, is that we have the unequal sharing of electrons because of that difference in electronegativity. A few slides ago, it said a subunit is a single polypeptide made of multiple polypeptides. A subunit is just, that was a typo if that said that, because a subunit is just a single polypeptide in multiple polypeptides. Like if there's multiple polypeptides, each one is a subunit. So if it said that it's a polypeptide made of multiple polypeptides, then that was just a typo. I'm not reading off the slides, so that is my bad. But yeah, a subunit is just a single polypeptide. So like if it's a single polypeptide, okay, good on that. Why is cysteine polar? And is there a way to know what's polar and what isn't? By looking at it, yes, that is a great question. So cysteine is polar because, or I think you mean why is cysteine nonpolar? I think that's one of the ones we fight on because there's a few that we disagree on that some people put in polar and some people put in nonpolar. Cysteine is one of them because you see the SH here is what I think we're referring to of why wouldn't that be polar? And I think that's because it acts as a nonpolar. It makes hydrophobic interactions. It doesn't make those hydrogen bonds like the other ones do in nature. So the electronegativity is probably just not as great. But it's one of those ones that's on a gradient. So there is a gradient, and I don't know if Professor Francis has showed you yet or not, but it is like most nonpolar to like least nonpolar, least polar to most polar. And it's kind of like a gradient. So cysteine would have been one of the less nonpolar ones. It'd be on the cusp versus something like Phenylalanine, which is a ring of C's and H's, which is probably the most nonpolar because that's insanely nonpolar. It's all C's and H's, and it does not want to be around anything. So that is a great, how to identify is a great question, because that's kind of what the goal of this class is to be able to look at these things and see. If you see a lot of C's and H's, it's going to be nonpolar. C's and H's have the same electronegativity, even sharing of electrons. They don't have any dipole movement, so they are as nonpolar as they come. If you see a charge, it's obviously ionic, and polar is going to be things like OH bonds, NH bonds, CO bonds, things that have differences in electronegativity. So things that are negative, so O's and N's, they are more electronegative than C. C is very neutral. And that means that the electrons are going to be pulled more towards the O's and the N's. That is what causes the polarity, is that the electrons are surrounding those O's and N's more than the C's. So if you're looking at something and it has, like, asparagine has this amino group and this double bond O, your electrons are, it's very obvious in this case to me, and hopefully one day it will be to you, that wow, there is uneven sharing of electrons. We know that in some cases amino groups are even charged in certain pHs. In the case of asparagine, it's not, but we know that that group is more electronegative because it can be charged in certain cases. So you look at that and you're like, wow, so there's different electronegativity, that's definitely polar. Versus you look at isoleucine up here and you see only C's and H's, yeah, that's definitely nonpolar. Methionine will probably throw you off if you see that S, but if you look at it, there's definitely more C's and H's. The S is in the middle. It's not going to be as easy to interact with anything, and it's not electronegative enough to make a difference. And then cysteine, like I said, is one of those borderline ones. And I think actually there's been a question in the past of why is cysteine on the cusp? Why do they sort it differently? And that's because it's behavior, and it's not very polar. It might be a little polar, but it's definitely a gradient. And that's the tricky thing about bio, or it's not just memorization. You look at it and it's like, huh, so not all nonpolars are created equal. Does that make sense? Does that help kind of understand? Okay, awesome. And then, so we talked about our polar charged, our polar uncharged, and then our nonpolar groups. We kind of just went into this, is that lots of C's and CC's and CH's because they have equal electronegativity. We call them van der Waals, London dispersion forces, hydrostatic interactions. There's all kinds of names. I always liked van der Waals. I thought it sounded cool, so that's what I wrote. Hydrophobic interactions, it's totally fine. Just pick what you want. I think in this class we favor van der Waals, as in just that's what we use the most. But yeah, so those are your interactions, and those are probably the most important thing to get out of this review session to know, is your different interactions between R groups, because that is the bread and meat of protein folding. Can people see this slide, or is it just black with weird squares?
It's black for me.
Okay, wait, what just happened? Oh, I'm going the wrong way. Now can we see it? Yeah, good. Okay, cool. I don't know what just happened. That was weird. Steric hindrance. This is your bulkiness and repulsion. This is what we were talking about. So this was a big question people had on our problem set. So what's more likely to be found on the inside versus the outside, or stuff like that? This is where we're looking at what can really affect your R groups and the interactions between them. So you look at glycine. Glycine is literally just an H as the R group. That is super small. So it's not really interfering with anything. Imagine like you're in a big room full of people. Like if you put a kid can squirm through, right? They're tiny. So they're not really taking up a lot of space, and they're not making a lot of strain. So it's more flexible around here, right? Because there's no strain. There's no, like if something's positively charged, that feels really like, that does not want to be near other positively charged things. It really wants to be near negatively charged things. This H is pretty much as neutral as you can get. So glycine is the least offensive when it comes to folding, repulsion, bulkiness, versus proline, which is cyclic and cycles, is just, it's simply bigger because of the direction or just the shape. You can imagine a line is thinner than a hexagon, you know, I'm trying to do hexagon, whatever. So there's a lot of steric strain for this because it has a shape and it's staying in that shape. So you can't, it's like trying to fit, has anyone ever tried to fit something like too wide through a doorway? It's that same thing. Is that like, you have to just find the correct way to move it. And so you probably just shouldn't put it through the doorway. So the backbone is more rigid because it has to be in this certain thing. It's like trying to put a metal sheet, like trying to bend a metal sheet versus trying to bend paper. It's more rigid, and so because it has to be this certain shape. Lots can form a helix by themselves. Yeah, so an alpha helix, I guess, can be formed by this. Proline kind of messes with me. Proline's definitely a really interesting one that can do some interesting stuff. You don't need to know a ton about that though. Phenylalanine is a carbon ring, which means it's super stable. It's super steric strain. Same thing. Backbone is more rigid. And it's the same reason as proline. It does not form a helix by itself, but yeah, you see these rings, these cyclic carbons, and they're definitely more steric. And so when you're comparing R groups, because sometimes it's like, if I switch, say I put in a phenylalanine instead of a glycine, is that going to affect folding? Well, you think about it, and glycine is way, way, way smaller. So if you throw a phenylalanine in, even though they're both nonpolar, that could definitely affect folding, because imagine your H is in this tiny little area and it just fits in, and then you try to put in phenylalanine, which is like huge. You try to put it in the same space, it's not going to work. So these are the kinds of questions that you're going to be asked. So this is a good thing to think of is size, and not just is it polar or non-polar. Size and steric strain are really important too. There's a lot of factors that you can play around with, which is why multiple choice is awesome, or not awesome, why open response is awesome, because then you can explain anything you want. R group interactions and tertiary and quaternary, I think we already went through this. I think we're feeling pretty good about this, I think, right? Is that your polar charged makes ionic, your polar uncharged make hydrogens, your non-polar make van der Waals. R groups, so they're hydrogen bonding in the secondary structure. Remember, we talked about the alpha helices and beta sheets are hydrogen bonds. The R groups aren't involved in that. So your hydrogen bonds are in secondary and tertiary, but they're different because your hydrogen bonds in secondary are between the backbone, and your hydrogen bonds in your tertiary are between the R groups. And at the bottom it says strongest to weakest. I think that might be a question for you all to answer. So does anyone want to either say it out loud, or can some people throw their answers in the chat of what the strongest bonds are to what the weakest bonds are? I'm going to throw, these are your three options, and I'm going to throw covalent in there too. So I have these three and then covalent. Can anyone rank them strongest to weakest? And if you get a correct answer in the group chat, I will read it out loud. Is anyone else weird that they drink tea and water at the same time? Okay, so I have a few disagreements. Okay, so it looks like we have some disagreement. So we did have one correct answer, and that is covalent, then ionic, then hydrogen. And we didn't put van der Waals there, and then van der Waals, yeah. So it looks like some people are thinking that hydrogen is the strongest. I would say hydrogen, it looks like the most frequently used here, but it definitely is not the strongest, because remember, those peptide bonds are covalent. Those peptide bonds are holding together that primary structure. Covalent is sharing electrons versus donating electrons. Covalent, like, you are stuck together. You are not getting split apart. So covalent are definitely your strongest. And then you're ionic, because the charge makes that unequal sharing of electrons, or that sharing of electrons, really strong, because we have those extra electrons or lack thereof. And then hydrogen bonds, because we have that dipole. So the dipole is weaker than the ions. It's weaker than the charge because it's just a partial charge. And then our van der Waals are, like I said, those aren't even really bonds, they're just interactions. So the order is covalent, ionic, hydrogen, and van der Waals. And it looks like a few people were confused on that. So I'm going to keep going just for time's sake, but we definitely, if you have questions about that, save that. And I definitely will stay on to answer those questions as well, if that is still confusing to people. Disulfide bonds, like I said earlier, they only happen between cysteine molecules because cysteines have S's, and disulfide, you need 2 S's, which are sulfurs. And it is the only covalent bond in secondary or tertiary and quaternary structure. Trick question, if I ask you what structure covalent bonds are involved in, you can say primary, tertiary, and quaternary, because cysteine disulfide bonds are covalent bonds. The only thing you'll never, ever, ever see a covalent bond in is secondary structure. You're not going to see disulfide bonds a lot, but they happen and they stabilize. And fun fact, I think I read this somewhere that the curlier your hair is, the more disulfide bonds you have. So I think that the ringlets in your hair, curly hair, is due to disulfide bonds. If I just made that up, I'm sorry, but I think I read about that one semester for an assignment I was doing. So if you have curly hair, you have disulfide bonds. Fun fact. Bond summary. This bottom part is probably going to be your most important thing. So I'm going to send this out, remember, but this is your bread and butter. This is what you really need to know, is what is involved in which structure. That's a big one, is understanding your bonds, but also knowing when you're going to see those bonds. You're definitely going to get a question about that on problem sets and on exams and everything. You're going to need to know. That is fundamental for the rest of the course. So that is definitely something that I would make sure you understand. And then obviously just what the covalent bonds are. We have our peptide bonds and our disulfide bridges, and then everything else is non-covalent. So that's our bond summary. Like I said, I will be sending out these slides at the end of the session. So don't worry about scribbling down notes if you don't have time, as you will get these. How does a protein fold? I think we're getting to, oh no, that was a long list. How does a protein fold? This is a big one, is what are you going to find on the inside and what are you going to find on the outside? The green things are the non-polars, and all the other colors are polar. If you look at this picture, your green ends up on the inside. Non-polar is hydrophobic. They're scared of water. They don't want to interact with water. So we're going to hide, essentially. Remember, this is 3-dimensional. So there's a pocket on the inside. Once we fold, you're going to find all your hydrophobic things hanging out in there, your non-polars. Your polars and your ionics are hydrophilic. They like water. They want to interact with water. So those are going to be on the outside. So that's the fundamental of where you're going to find the groups, is that you're hydrophobic, you're going to find on the inside. You're hydrophilic, you're going to find on the outside. Like, end of story. That's what's going to happen. So that's pretty much all this is showing you. And I know there's some problem set questions on that, so I won't be surprised if we get a few of those by the end, but yeah, that's what's going to happen there. How to tackle amino acid problems. These are tips and tricks from the TAs over the years of how we think that it's most efficient. I'm just going to summarize this. I'm not going to read it word for word, but a lot of times you have to compare amino acids. So you're going to get your chart. Don't worry about memorizing it. But a lot of times you're going to have to compare amino acids. So start with describing their different sizes, their chemistries, how they differ between each other, and what bonds they can make. So if you see a polar and a non-polar, okay, let's compare the size and the chemistry, but also polar can make hydrogen bonds and non-polar can't. So there's a big difference as well. And then arguing, basically a lot of times it's going to be like, is this going to change the structure or not? So you make that argument. Yeah, because it's bigger. Yeah, because it's polar when originally it was non-polar. No, because they're essentially the same. Because, you know, if you look at glycine and alanine, they're very similar. So maybe that one. Sometimes it's okay to say, maybe it would, or it could, but maybe it won't. It might be fine. The point of these open responses is to get you to think and argue your point. So if you're like, yeah, this is going to change, tell me why. Why is it going to change? What changed? So you look at your size, your chemistry, the types of bonds you can make, and you start there and you decide, okay, this is definitely different. If it's different, if it's making different bonds, that's a big deal, probably, right? But if it's pretty similar and it's just like a little bigger, like it has an extra CH, then maybe it won't affect it. In reality, anything can affect any kind of folding, any mishap, but some are going to be more likely than others. There's some practice problems down here for you. I don't think anyone wants to type this out right now or speak up necessarily. I know this is being recorded. But if you want to do this practice and you want to email me the answers and have me check them, or just have it for your own sake, that is awesome, including the YouTube videos. Just if you're going to email me and ask me to check your answers, if you're on the right track, let me know what you're talking about, because I don't, like, just let me know what the question is. I think that's a good way to do it. I think these are great practice for you. So yeah, that is some advice on how to tackle these problems. Problem solving approach. So tyrosine and phenylalanine, they're very similar in size, but that tyrosine has that OH. So they differ in that chemistry. We can make a hydrogen bond with tyrosine. So yeah, that probably would change, you know? I don't know what cysteine we're arguing against. Yeah, so cysteine, remember we talked about the nonpolar, it's slightly more polar than the other nonpolars. So sometimes that can be tricky, but we talked about that already, so I'm not going to delve back into it, but this is a written out explanation of what we talked about earlier, is that it's slightly more polar, but it's still nonpolar, so it's one of those tricky ones. So it's a good one to talk about. If it's thrown on an exam, talk about that for sure. And then here is our beautiful chart that you're going to get. You can find it on Google. I use this chart still to this day. It's my favorite thing ever. You will definitely see it a lot. Don't necessarily get dependent on the colors because exams are in black and white, but yeah, you will definitely get used to this chart. Protein modeling, we'll go through real quick. I don't know why Syed's name is on the bottom. I just want to let you know that was a TA from last semester. I'm not quite sure why his names are on the slides randomly, but here's our different ways to model. We got our ball and stick wireframe. We have some pictures, but this shows your backbone in your R groups. The balls are the atoms and the sticks are the bonds. Wireframe just doesn't have the atoms necessarily, like it just has the bonds. So if you want to show your backbone and your R groups, this is a good one. Space fill shows your overall shape. So if you want to know what your shape looks like the shape is of the protein, like what it looks like in three-dimensional space, you're going to use space fill. Ribbon, you can't see your R groups, you only see your secondary structure, so that you see your helices and your beta sheets. Those are the prettiest in my opinion. Your backbone just shows your backbone, no R groups. And you can definitely overlay models on top of each other. If you overlay space fill, you're probably not going to see much, but if you have wireframe and ribbon model, then you probably are going to be able to kind of see that a little better, see a little more. Try to come up with pros and cons of each. For example, space fill model, you can't see your R groups, so you don't necessarily know how it would interact with something else. Ribbon model, you can't see your R groups, but for some of the ones that you can see your R groups, maybe you don't know if it's an alpha helix or a beta sheet and you want to know that. So they're all good, just depends on what you're doing. And here are the beautiful pictures, your ball and stick model, which makes my head hurt. Space fill, you probably are definitely used to if you've taken Gen Chem. You get those pictures a lot on owls. Ribbon model, which you will see a lot in this class. Ribbon model, like I said, is my personal favorite. I think it looks the coolest. And then the backbone model, which is like the knockoff ball and stick model. That's a joke, they're different. But yeah, that just shows the backbone. So here's some pretty pictures that are definitely helpful. What affects protein folding? Environmental factors. So we got temperature, pH, UV radiation, all those things. And these affect because if it's too hot, things can get messed up. Bonds can break. Heat can break bonds. And if the pH, if it's super acidic, it can break bonds. So if you change the environment of a cell, you can mess up the folding. So if it's misfolded in any way, then it's abnormal because structure equals function. You mess up the structure, you're messing up the function usually. So yeah, the best question, the one that I got wrong probably in every bio class I ever took, from 151 to 152 to Selma, is the egg white question. I don't know if anyone's seen it, but that's a great example of this, is when you break an egg and you heat it up, why does it turn white? The heat actually is breaking the bonds, the tertiary structure. And because you have hydrophobic on the inside, they freak out because you just broke them and you expose them to the water. So they all clump together because they aggregate because they want to try to hide. So they all just hang out together. And that's why the egg turns white, because you get a bunch of aggregation of these hydrophobic groups. And yeah, that's that. So if you ever want to think about proteins clumping next time you're eating an egg that's cooked, knock yourself out. But yeah, that's why heat affects eggs and turns the eggs white when you cook them as opposed to clear. So that's an example. I hope that sticks with you. And I really hope Professor Francis asked that question this semester so that the 23 of you here can get it right. How are the bonds affected? Like I said, this is all kind of very redundant. I kind of touched on this earlier, is that anything can affect a hydrogen bond or an ionic bond. They're weaker, so heat, pH, everything. Covalent bonds are very hard to break. So you kind of got to throw like hydrochloric acid on that to really break it. Apparently, if we're unsure about a particular environmental factor on an exam, just ask us. We're not on exams yet, so I won't worry about that. But most of the time, the environmental factors, you don't have to worry about it breaking the covalent bonds, just because like that doesn't happen a ton in nature. Like that's very severe. So like that happens in a lab. Like we can do that in a lab, but if we're assuming the change of environment in natural cause, just assume that the covalent bonds are still intact, because those are a lot harder to break. You're rarely going to break them. Okay. It says Coachella bonds. Sorry, in the what are they called? Captions. That was funny. Heat causes unfolding. Oh yeah, so the Goldilocks hypothesis. More thermal energy equals more stable bonds, but more stable bonds are not always good, because sometimes you want your bonds to be able to break. You don't necessarily want something to stick together forever, you know? Like you want things to stick together and come apart and stick together. Some things dimerize, so they come together and break apart. So sometimes it's not just right. That's the Goldilocks hypothesis. Sometimes it's not just right. So don't assume that just because it's covalent, it's better, it's stronger, which you need in some cases, but some cases not. So that's a good thing to remember, that we're not qualifying this as better or worse. It's strength, but sometimes we really need those weak interactions. That's good in a cell. We need that because our cell is very transient. Things are changing a lot at all times. So it's very fluid, not transient. I would say it's more of a fluid process. pH and salt affects charges. So like I said, heat just breaks things. You can affect charges. So pH obviously is salt and affecting the charge. That's what pH essentially does. So cells have a certain pH. I think it's around 7.5 or something, like a little, it's around 7, which is neutral. That's not true. I don't know. I take that back. I'm not quite sure what the cell pH is exactly. So yeah, so we control that, but if you just add like a ton of salt, like you can probably make a cell explode. And then high energy wavelengths, cancer. People know that you can get skin cancer from the sun, and that is because light, these UV rays can affect 7.4. Okay, I wasn't as off as I thought I was, 7.4. Yeah, I don't know why I suddenly like freaked out and thought that I forgot what the pH of a cell is. Yeah, I do believe it is around 7.4, 7.5. Thank you, I feel a lot better now. I was like, wow, I'm giving misinformation. But yeah, point is, cells are not neutral. But yeah, wavelengths, this is your explanation for why, why people get skin cancer is that those UV rays can be really harmful. And it can break non-covalent bonds. Really strong UV rays and light can break covalent bonds. It's a lot harder, like like we said, that covalents are super strong. But yeah, that's a big thing for skin cancer, and why that's happening is because that UV rate is affecting your bonds in your body. It's crazy to think about. So wear sunscreen. So that is it for my presentation. Does anyone have any questions not related to the problem set, related to the slides, that they want to ask before I stop the recording? And if you want to ask your question after I stop the recording, that's also fine, but this will be uploaded to YouTube. So any other questions that you want to ask before I stop the recording? I'm going to wait for a second because I know sometimes typing it out takes a minute. Okay, I don't see anything in the chat, so I'm going to stop recording now.